Thanks a lot for that kind introduction, also for inviting me to give this lecture. It's always nice to be back, and I have some special connections to this area. So my grandfather used to live in Schluchse. I used to be here every summer, so it's nice to be back, although it's almost half a century since I used to come every summer. But it's a nice research community, and I've heard a lot of interesting work ongoing today. So thank you very much for that. What I want to do today is to introduce you to some of the work we are doing in the lab in Norway, and I will focus, as the title says, on the neural codes for space and time. But before doing so, I will start a little bit more broadly for those who are not neuroscientists here. And let me then begin by addressing the complexity of the brain. So in the human brain, there's approximately almost 100 billion neurons. Each of them have connections or synapses with about 10,000 other neurons. So we are talking about connections of approximately 10 to the 15th. And this is where we believe most of the information is in the brain. So it's very small, it's very distributed, and extremely hard to access. So how can we do this? This is not an easy task, and this is also the reason then why for those who wanted to understand cognitive functions, emotions, behavior, the brain was for many years treated as what was often called a black box. I mean, psychologists tried to avoid it simply because it didn't seem like it was addressable. But things changed, and very much started to happen in the late in the 1950s. And I think one of the developments is the gradual breaking of the barrier that has traditionally been between, on one hand, physiology, studies of how cells work together in the body and in the brain, and psychology, on the other, the study of behavior. And as I said, psychology developed for the first part of the previous century, essentially without considering the brain and its structure or dynamics to any considerable extent. But this changed, and on this slide here I put up a picture of David Hubel and Torsten Wiesel, who are one of the founders of modern neuroscience. So what they did, starting in the late 1950s, and with some highlights of their work in the 1960s, is that they showed for one system of the brain, one of the sensory systems, or the outer parts, the interfaces of the brain with the external world, namely the visual cortex in their case. They could have done it in other sensory systems, auditory cortex, olfactory cortex, and so on. But they used the visual cortex as a starting point, and then found, as is shown to the right here, that there are cells that respond specifically to lines or bars, stripes of certain orientations. So you can see, for example, this cell is very active. Each vertical line illustrates a signal or an action potential from this neuron, and you can see that it responds very strongly to the vertical or almost vertical line there. So this was the detection of the earliest elements for sensation in the brain, and then by implication also the beginnings of cognition or thinking in the brain. So the earliest building blocks, and that introduced really a major change. Of course, it was not only their work. I use it for illustration, but it was very influential in starting to bridge physiological and psychological approaches to the brain. Torsten Wiesel is still around and is celebrating his 100th birthday now next year, so there's already a big celebration in planning, and it's very fun that I'm going there and participating in this next fall. So, so how does this interact with the rest of the brain? So here you have a schematic of the visual systems of the brain, the visual parts of the brain. So each box is a certain brain area, and they are connected with lines that roughly illustrate the strength of the connections between the areas. The bottom is where you have the eyes and the sensory, the visual inputs get in through the retina, the thalamus, and into the brain. And the purple area here is the first visual area where they did their recordings. Now, this is in a way the bottom of the brain, the interface of the brain with the external world. But having a training as a psychologist, I was actually more interested in the top of the brain, and that's what you have here. So if you focus on the same diagram, you now see an arrow at the top. Because the functions I was interested in, cognition in general, thinking, planning, decision-making, memory, they all were involved many brain systems, and certainly not only the ones that were in the outer parts. So I had a special interest in space and time, which we worked on in our lab for many reasons, but one of the reasons is that it is far away from the sensory input, so that you can actually use these systems to try to understand the operations of the brain in the absence of sensory inputs. So it could be used as a way to find the rules, the algorithms that the brain is using when it's trying to make sense of things. And that's why I now put up this picture of Immanuel Kant, because as you all know, what he proposed is that how we perceive the world is very much dependent on a few constants in a way, if you can call it that, like our perception of space and time as examples, which we come with when we enter this world, and which are, at least today, you would say, he would have said that it's functions of how the brain is wired up, and we cannot really escape it. So this comes prior to experience, and is a way, a function of how our mind or our brain is set up. So whether we want it or not, we have to perceive the world through these glasses, and addressing the mechanisms of space and time could then be an interesting way to try to understand how the brain works in a more general sense. And for most of this talk, I will talk about space, because that's where most of the work has been done. I will also address time in the second part, but time is more nascent, we know less, and although it's so pervasive, part of everything, then it is a little bit harder to understand. So let's begin with space. And where is space? Of course, in a sense, the entire brain is involved in such a complex function. With space, I refer to our ability to know where we are and find our way in order to find food, find mates, and so on. So you can imagine how fundamental this is for survival. If we hadn't had the ability to imagine our location in the environment and finding our way to other locations, we would be lost and we would not survive. So it's very fundamental. And now there are some brain areas that seem to have a special role, in the sense that in the human brain, if they are damaged, then we lose the ability to know where we are and get totally confused. We get lost and don't find our way back. And one of these areas is the hippocampus, which is shown here in red, and the associated area of the entorhinal cortex, shown in blue. So they are both part of the cortex. The cortex is the sheet that overlies the brain and accounts for very much of our intellectual activity. So the hippocampus and the entorhinal cortex are actually, they are in the medial temporal lobe on the sides, but actually folded under as the brain or the cortex curves in. So you don't actually see much of them, and you don't see the hippocampus at all from the outside, but it's still part of the cortex, an old part of the cortex. So it has been known for more than half a century that the hippocampus has a special role in memory formation. So if the hippocampus is damaged, then what happens is that you don't remember any daily life experiences, and along with that, there's also a loss of the sense of location. The entorhinal cortex has not been addressed or investigated much recently, so it's only during the last 10-20 years that there has been a much stronger focus on it, despite the fact that entorhinal cortex actually connects the hippocampus with the entire rest of the cortex. So it's an interface in many ways, in addition to doing its own things. So it would be, for anyone interested in how the hippocampus works together with the rest of the brain, one needs to go to the entorhinal cortex. So, but this is the human brain, and the human brains aren't all that easy to study. So for that reason, much of the work in neuroscience is done in animal models of various kinds. So you start from the right here, see the human brain with its almost 100 billion neurons, and you go through monkeys and you get to the rat with about 200 million, and you get to the mouse with about 70 million. You can continue, you get to the fruit fly, Drosophila, with 135,000, and you get in the end to a small nematode of the size of a comma, which has 302 neurons, and is used as a model organism for understanding brain circuits. But sometimes you don't want to go all the way to the simplest systems, because even if you can use those simplest systems, there are some functions they don't have. They probably don't think all that much, and memories at least are very simple, and so on. So if the starting point is that we want to understand our sense of space and time, which are fundamental parts of the mammalian brain, then we have chosen, and much of the work is done in mammals, and especially in rodents, in rats, and more recently much more in mice, which have these abilities, and where these abilities are strongly developed. So rats and mice are extremely good at finding their way, and it's also easy functions to measure. So this is all a function of evolution, and what you see here is an evolutionary tree for mammals, starting from the bottom left with a common ancestor, and then branching off where you have humans at the top right, and you have rodents, for example, in the middle or the left side. Sharing, then making it possible to assume that much of the circuits is similar, and that's also what you see when you study the brain circuits and see that they are actually built up very much according to the same principles, although humans have so many more neurons. The underlying principles are very similar. So with that as a motivation, then, much of the work has been done in rats, and as I said, more recently, much more in mice. And that applies also to the studies of the brain's spatial systems, systems for mapping location and for navigation. So this is a milestone in that research was in 1971, when John O'Keefe used quite recently developed microelectrodes. They were actually similar to the ones that were used by Jubilee Wiesel, and which I showed whom I showed in the beginning. But he used them to go into the brain area of the hippocampus. At that time, it was known that hippocampus was important for memory formation. But what O'Keefe observed, somewhat by accident, was that neurons in that brain area actually encode location of the rat or the animal. So I'll illustrate this now with recordings from a rat. So you see the rat brain at the bottom left here, and you see an arrow pointing to the hippocampus in a rat brain. And the red one area is the hippocampus in a human brain. So they look a little bit different, but they have the same sausage-shaped structure, and the internal organization is very similar. So this video that I'm going to show very soon is a video of a place cell to illustrate the concept of a place cell in the hippocampus. So what O'Keefe did, as I said, is to record activity using microelectrodes, very small thin wires of approximately 20-30 micrometers in diameter, which were uninsulated at the end, so that he could, at the end of the wires, could pick up electrical activity from surrounding cells. And that enabled him to record activity from the hippocampus in a rat while the rat was running around freely in an environment, and then connected with a cable towards for the occasion connected with a cable to the microelectrode on the skull. So when I start the movie, you will see the rat running around chasing chocolate pieces, because the rats like chocolates, and that's just a trick in order to keep the rat running around and visiting every possible place. And you will be hearing a sound, a bop sound, each time that cell is active and fires an action potential, a signal from that neuron. And you will also see a red dot when the cell is active. So let's start the movie. And you can now hear and you can see that this cell is active specifically when this rat is in the upper left part of the box. And otherwise it is silent, not sending any signals to other cells. So this area where the cell is active was called by O'Keefe a place field, and the type of cell was called a place cell. And you can also see it at the bottom in a heat map, which shows the box seen from above, and in red it shows where the cell is active, in blue it shows where it's not active. So different cells in the hippocampus had different location preferences, so together they all filled out the box and more than that. And this led then O'Keefe, shown to the right here, and Lynn-Nadel in 1978 to suggest that these cells are part of an internal map of in the brain. And they built then on the concept of internal brain maps that was proposed almost more than 30 years earlier by Edvard Tolman, a psychologist, but at that time Tolman didn't have access to the brain, so he introduced the concept but had no idea about how it could be implemented in the brain. So these are maps of the external space, but maps that also include information about what's going on at different places in the space. So now then, in the hippocampus, quite in the middle of the brain, far away from the sensory inputs, there are cells that encode location. And then these cells were studied in the 1970s, 80s, and then in the 1980s, and with a publication in 1990, another group found another type of cell that also, again, by coincidence. So Jim Rank to the right here and his student Jeff Taube recorded activity from a neighboring brain area, now called the presubiculum. And they were actually hunting for place cells in the hippocampus, but these microelectrodes got off the normal track and went into the wrong area, and suddenly they found that there are cells that respond not to location of the rat, but to the direction it is facing. So we call these cells head direction cells, because they signal the direction of the head in the space. And this cell that you see here is active only when the head is pointing in their reference frame between 240 and 300 degrees. And then when the head is in other directions, there's no activity at all. So then there is then a directional system in addition to a place system. So how are these linked? Well, that remained a mystery for a long time, and to some extent it's still a mystery, but not so much. But the years passed, and in 2005, then our lab started to record from the entorhinal cortex. Entorhinal cortex is then the blue area, which you see in the rat brain in the cartoon up here, and it's also the area shown here in a rat brain seen from behind, the colored area, the upper part, which is called the medial entorhinal cortex. And in there are also cells that respond to location, which you see in the video. So you see the rat running around again, but this time the cell, this one particular cell, is not active at one location, but at many locations. And you can see how those locations actually form clusters in the local space, and clusters that are quite regularly organized. You can also see that on the plot to the right, where in gray you see the path of the animal when it's running around in a box that is 220 by 220 centimeters, and it takes half an hour to visit every possible place in the box. And then in black you see where that particular cell is active when the rat is running around. And again you see clusters of cells, and you see that they form a particular pattern, so you can tile the pattern with equilateral triangles, and you get a pretty good representation of the space. So it's a pattern that consists of equilateral triangles, or if you like, hexagons, very regular hexagons that tile the entire space. So this looked like a grid that covered the available space, and for that reason we call the cells grid cells. And it seemed from their very firing properties that they contain information both about location and direction, and you can easily put that into a computer model, and with information from different cells that have different frequencies of activity, you can infer the location of the animal at any given time, just as you can do from place cells. But then it turned out that these are not the only cells in this brain area in the medial entorhinal cortex. Actually, the grid cells, while they are the most abundant ones, they also mix with a number of other types of cells, and one of those other types of cells we discovered in 2008. So these we called border cells. So those cells are active only at the borders or boundaries of the environment. So this cell that you see here is active on the right side of the box, and that happens regardless of whether the box has a horizontal shape or like a horizontally lying rectangle or a vertical one, and also if a wall is inserted into the environment. So this particular cells always fires along a wall in the environment, and other types of border cells tile other walls, and together they give information about where the local environment ends or meets a boundary. So completely different from grid cells, and a grid cell could never be a border cell could never be a grid cell. They're totally different types of cells. And then other types of cells came, so in 2019 we found that there are some other types of cells that we called object vector cells. These are cells that are active with reference to local objects or landmarks in the environment. You see such a landmark here in this box, and you can see this is a Lego tower placed anywhere in the box. And it turns out that there are some types of cells that when you introduce this object, these cells start to fire either at the object or at some distance away from it. And when they fire some distance away from it, it is typically on the side and some distance, in this case maybe 20-30 centimeters away on the west side. And if you then move the object, then the activity field follows the object. So that you can see at the bottom here. So here you see an object fairly in the center here, and then an activity field on the north side. Then you move the object, the ring here, to the southwest part, and the activity field is still on the north side, some distance away from it. So that these cells encode vector relationships to prominent landmarks or objects in the environment. A completely different way of encoding location, but you can also use these cells to decode where the animal is at any given time by looking at the combination of activity in such cells. And they are also in the entorhinal cortex, quite overlapped with the grid cells, although maybe not entirely, a question I will come back to at the end. So this is, and in addition, are cells that, as I said, encode direction, and there are other cells that encode speed. So it's a mixture of cells with different functions. You may wonder if those exist only in the rat or the mouse brain, but that's not the case, because they have also been discovered first in bats. So bats are on a completely different branch of the phylogenetic tree for mammals. So the fact that they have grid cells, head direction cells, place cells, suggests that the system is actually quite similar and must have evolved early. And then there are grid-like cells also in monkeys and humans. In humans, they are recorded typically from patients with untreatable epilepsy, where the patients get electrodes implanted, similar to the ones in the rats and in the mice, in order to identify where the epileptic focus is and where the seizures start, so that you can access and possibly remove tissue. So then you get for free recordings from those cells while the animals or while the patients move in space, typically in a virtual environment. So, but that's a beginning, but not enough. So we are now at the point where you know that in the entorhinal cortex, in this area, medial entorhinal cortex, there are a number of types of cells that have quite specific functions in representing or encoding specific aspects of the external spatial environment. So I mentioned grid cells, head direction cells, object vector cells, border cells, and place cells in the hippocampus. But how do they all work together? So that's really the million-dollar question. You need to understand how they work together if you want to understand how we can have a sense of space. And this has been quite inaccessible for a long time, simply because we didn't have the methods to record activity from a sufficient number of cells at the same time during performance of behavior. So this has now completely changed, and it's due to, at least in part, to the development of quite new technologies. So first of all, the electrophysiological recording technology has completely changed within the last 5 years. So as I said at the beginning, and when we did our work on grid cells and all the other cells, we used conventional electrodes, which were small wires that were uninsulated at the end, or you can pick up maybe 5 or 10 cells around the end of the electrode and then figure out when were they active. The new technology consists of very small chips that are the width here of 70 micrometers, and are then like rods that can be inserted into the brain instead of the small microwave wires. And then on these silicon probes, there are uninsulated recording spots all the way along at distances of 15 micrometer in this case. In the newest version, this is an older version with 20 micrometers, close enough that on each recording spot you can pick up the electrical signals in the vicinity. And then if you accumulate the signals that you pick up in parallel from a choice of more than 5,000 channels now, then you can at least now record 384 of them at the same time. And in a year you can record 1,536 at the same time. So it's really growing fast, and it means that you can, where you a few years ago could obtain on a lucky day you got 5-10 cells. Now you get, on a bad day, you get 1,000 cells. So it is a complete change of access to the neural activity in the brain. So using these electrical neuropixels probes, as they are called, we were then able to get some of the first insights into how the grid cell system works in terms of population activity, in terms of many cells working together. And what you see here to the left is an example of a recording from grid cells in an experiment with 2,460 cells, out of which 483 were determined to be grid cells, and out of those, again, a subset of 149 were cells from the same module of grid cells. And here I have to say that grid cells come in clusters or modules of cells that are quite similar, so they have the same spacing of the periodic pattern, for example. And in one of these modules, then we, as I mentioned, we recorded 149 of them, and you can see those here. So each square here is the recording from one cell. So it's a map similar to the ones you saw. So yellow here means high activity, and blue means low activity. And you can see that all of these have this periodic hexagonal pattern. So they are grid cells, all of them. This is the work of Richard Gardner, who was a postdoc in the lab and still is, and Erik Hermansen, who is from the mathematics department. And you will soon understand why this is a work that we do together with the mathematics people. So then we have to look at the plot to the right. So the plot to the right shows activity over a certain time, approximately 5 seconds, which is the time from left to right here. And each row here shows the activity of one neuron over that time. So black means when it's active, and white is when it's not active. And you then see 149 lines with the activity of all of the cells. So what we are now looking for is not the activity of the individual cells, but it is the activity, the combined activity of those 149 cells from the same cluster of grid cells. And we can do that by looking at combined activity in the time slot, for example, shown with the red column here, and then ask what is the combined activity at different times. And in some sense, that would be a hard task, because if all of those 149 cells were independent, in order to show the changing activity when the rat is walking around in the box, you would actually have to plot the activity in a 149-dimensional coordinate system, which would at least not be very good for visualization. Would be really hard to see anything at all. But we are so lucky that in this brain area, in most brain areas, but particularly in this brain area, the cells actually turn out to be highly correlated with each other, so that you can predict the activity of other cells from a subset of the cells. So you can actually break down the activity from the 149 dimensions down to just a few dimensions, and you can still explain much of the activity using dimensionality reduction techniques such as principal component analysis, PCA, which is a very old technique, but also there are many more newer forms that are not linear, don't rely on linearity, that are used for the same purpose, and for this they provide just the same. So what happens if we now break down the activity to 3 dimensions or 2 dimensions? So that's what you see here. So you see the activity now as a point cloud. Each dot in this cloud, if you look at the left one first, each point shows one time point in the recording. And all the time points together forms the cloud, so that you can see where the activity moves around. And you can see that if you break this down to 3 dimensions, if you plot the dots in a 3-dimensional embedding, you can actually see that it forms a very recognizable shape, namely a torus or a donut. And you can see in color from yellow to white, yellow to red, you can see the activity that took place during 5 seconds when the rat was walking around on a certain path in the box. And you can then see from the illustration here that the activity actually moves around smoothly along on this torus. It always stays on the torus, but moves to different places on the torus in accordance with the animal's movement in the environment. So in a way, this is an internal map that reflects the animal's position in the environment. When the rat moves to a different place, the activity on this torus also, the combined activity also goes to a different place. When the animal comes back to where it started, then the activity on the torus is also back to where it started. So this is an internal map of space that you can see when you have access to the activity of so many neurons. But now you can say, well, these are the best examples. Sometimes it's more noisy, so how can you actually show that this is a torus? Because maybe in a more noisy recording, you wouldn't immediately see it. So that's where we need mathematicians and people who have worked with in the field of topology in mathematics. We have methods for counting number of holes in various dimensions. So this is, we then used a method called persistence cohomology to count holes in 1 dimension, in 2 dimensions, and in 0 dimensions. So for a torus, what you would expect is 2 holes in 1 dimension. So those are the ones that are shown with the red rings here. So there's 2 orthogonal circles. And in addition, you would expect there to be, in a torus, 1 hole in 2 dimensions, which is the inside, the 2-dimensional inside of the torus. And you would expect, it's not really important, but you would expect also 1 hole in quotation marks in 0 dimensions, which is just a number of connected components. So in this case, it's just 1 structure. So let us then ask then, what does such a counting lead to? So if we begin with a recording from an animal that is moving around in a box, like you saw in the video, you would actually sort of expect it from the periodic pattern that there should possibly be a torus if you wrap the top of the map with the bottom and just connect the sides with each other. So what you see here is a so-called barcode diagram, which shows the number of holes in 1 dimension, which you see in the middle here, H1, and the number of holes in 2 dimensions. So the long lines are the holes which really stand out strongly from the noise. And the noise is shown by just shuffling the data, which is shown in orange or brown in the background. So you see there are 2 holes in 1 dimension and 1 long line or 1 hole in 2 dimensions, as you would expect from a torus. Now the same happens if these animals walk in a different environment where you don't really see very nice grid patterns, but you still see this same structure. And perhaps more strikingly, you also see it when the animals are sleeping. So to the left, you see a recording from REM sleep, or this is when in REM sleep humans is when we dream, and slow wave sleep is the other type of sleep, and you can see the same pattern again, 2 long lines in 1 dimension and 1 in 2 dimensions, which then suggests that the activity moves around on this internal torus-shaped map, regardless of whether the animal is actually moving anywhere in the environment or not. It's just that this map is there and the activity goes around on the map, whether you're awake or not, or whatever you're doing. But then when you navigate, what comes in addition is that you actually apply this internal map and align it with the environment so that it matches the environment and is similarly aligned next time you get back to the environment. So I could have said more about the theoretical fundament of this, but one thing to emphasize here is that why I think the torus finding is important is not really because it's a fun fact in a way, but it is also important because it says something about how our internal maps are actually wired. First of all, because they exist regardless of behavior, it suggests that you have these maps wired in the brain independently of experience. The other thing is that it is circular. So imagine what if you had such a map in the brain, in the entorhinal cortex, for example, and you had a bump of activity shown there in red that moved around on the map according to where in the environment you are. Then you might ask, what happens when you get to the end of the map? Do you fall outside? So this is the end of the world problem that geographers had some centuries ago. And the solution is, of course, that you make it circular. You connect the ends both at the sides and at the top, and there are various shapes you can get. You can get a sphere, a ball, but a better solution would be a torus, because then you keep the spatial relationships from the environment in the torus, so that distances are preserved. And that has been suggested from theoretical approaches for almost 3 decades. So it is really work that went on in parallel in theoretical and computational neuroscience for a long time. But theoretical neuroscience and experimental neuroscience lived for a long time their own lives because there were no methods to link them. So this is why it's so exciting times now, that you can actually have the back and forth dynamics between experiments and theory, because you can actually test those models, and that also means then you can develop them, because very often they don't quite match reality. But the back and forth dynamics is extremely healthy for the science of our field, and it verifies one of the sort of models for how the brain is wired, because a kind of model that is called continuous attractor network models has suggested that you get these bumps of activity that move around on an internal map by wiring together cells that have similar spatial preference, or cells that are active in the same places, and then you inhibit connections between cells that are different. And then if you do that, you should get a bump of activity, and that bump of activity should move around. And if it moves around on the torus, you can actually replicate much of the data that you see experimentally. All right. Then you may ask, if this is so independent of behavior, do you have to learn such a torus, or is it actually there from the beginning? So Matteo Guardamagna, in a postdoc in our lab, has studied this, and it's still ongoing work, so it's not published. But he started out by recording from rat brains at the age of postnatal day 20, P10, 20, went to P15. That is, P15 is a magic day, because that's when the rats start exploring the world. They leave the nest from the mom and start to make loops out of maybe 30, 40 centimeters before they return, and they make these loops just like human children do, and they come back to the mom. And this is when they start to get experience about spatial relationships in the world. And it would be an interesting time to find out if they actually have grid cells. So this shows the recordings from approximately 100 cells that are the most grid-likes he could find, cells that he could find at that age. And you can see it's not really periodic. It's quite a noisy pattern. But when he did the mathematics on it, he actually found that, again, there are these 2 long lines in 1 dimension and 1 long line in 2 dimensions. So these data are so preliminary that it doesn't yet include the shuffle data, which has a control, but the new data suggests that actually there are these 2 long lines and 1 long line in 2 dimensions, suggesting that the torus is actually there, even if you don't really see it in the individual grid cells, because the grid cells are sort of drifting around and not very well anchored to the environment yet. So then he moved on to, skip that, to postnatal day 11. And 11 is when the rat pups are just sleeping all the time. I'll actually start a movie here of a sleeping rat. And it's a funny movie because you don't see anything, but the rat is actually sleeping. And you will see a twitch after a little while. But he recorded while the rats are sleeping. And at this age, the eyes are still shut. They are shut until postnatal day 14, and the ear canals are closed. So the brain lives in a quite isolated from the external world. And nonetheless, you get the same again, 2 lines in 1 dimension and 1 line in 2 dimension, meaning that these point clouds have the topology of a torus. It was a little bit more difficult to do this because you don't know in advance which cells are grid cells, but you can cross-correlate all the cells and then take out clusters of highly correlated cells, and then you find that some of those clusters have this topology, which probably means that these cells will become grid cells at a later age, but they already have the internal structure of being such an internal map and just has to be aligned with the world during the subsequent days. So finally, he did that at postnatal day 10, and there are no grid cells there's no clustering any longer. So at that age, there is a change in the combined activity of the cells. So you can see that on day 10 at the top and day 11 at the bottom. And you can see that there is structure emerging in the network activity that is there on the bottom one, where you can see bursts of activity, which is not there on the top one. But we don't know yet exactly what it is, except that there seems to be a major change in the way the cells behave together. So that is still to be determined, but for now, I think it suggests that the brain or the brain's position system has actually an internal map that can be used later for mapping the space, which is independent of major sensory inputs, such as visual inputs and auditory inputs, and nonetheless the activity moves around on those maps, which is quite consistent with the general view that Immanuel Kant suggested several hundred years ago about the idea that we come to the world equipped with a way to perceive the world in terms of space and time, and that this is both good and bad in a sense, because it's a huge advantage. That's very easy to then adapt that map to the world, but also prevents us from actually knowing anything about the world, what the world is like out there if there had been no human beings. But then I'm over to philosophy, so I'll leave that to others. But let me now ask at the end of this section, before I move on to the time part, then is there any role for experience? So we tested this by raising rats for the first 2 months in different environments. And one of them is shown to the left, is a spherical environment. The rats lived in 2 months in a sphere and did not really see anything other than the local bedding and food inside, and together with their siblings, and but no fixed geometric cues, or at least reduced availability of such cues. Others grew up in boxes with clear walls, so they had some structure, and yet others grew up in an enriched environment with lots of toys and climbing opportunities. And then we asked, did grid cells emerge in both of them? And let me also mention that all treatment of these animals had to occur in darkness. So Ingvild Ulsager Kruger, who did these experiments, she had to wear night vision goggles for all these experiments to see the rats and to handle them, so that they didn't get access to anything outside. So on the first day of testing, when they were put in a normal recording box at the age of 2 to 3 months, then the grid cells were more striking in the two last groups, but not in this, not very striking in the ones that grew up in the sphere, on to the left, upper left there. So you can see it's a quite noisy pattern. But this recovered very soon, and after 3-4 days, then after 3-4 days, then you get the grid pattern. So it means that these grid patterns develop regardless and despite quite extreme rearing conditions when they are not exposed to geometrical cues. So with that, then a few words about time. I want to mention 2 aspects of time, because time is very complex. There's many different things, but one function that the brain encodes is the duration of events. How long does something last? So how does that happen? And one way to address this would be to record again, now using NeuroPixels probe to record activity from many cells at the same time. And you can do that as shown here with some hundreds to thousands of cells in lateral entorhinal cortex, medial entorhinal cortex, hippocampus at the same time. And what you then do, this is the work of Ben Kanter, a postdoc in the lab, is that you can again use these dimensionality reduction techniques. If you have 1,000 cells, you don't have to plot it in 1,000 dimensions, but can bring it down to just a few dimensions with the same methods that I mentioned. So this is what Ben Kanter did, but the difference is now that he selected the dimensions that explained best or most of the variance in time, so which were most correlated with the passage of time. And first, in medial entorhinal cortex to the left and CA1 of the hippocampus in the middle, you could see that there's one dot of the joint activity at every given time. So this is a session lasting 10 minutes. So you see that encoded from light to dark. And you can see that those dots essentially are at the same location in this plot in state space. One component on the x-axis, the other component on the y-axis, and essentially just moves around on the same space all the time. This is very different from what happens in the lateral entorhinal cortex, because there you can see that the activity actually evolves gradually through the state space, beginning here to the left and then going as the rat is walking around over those 10 minutes towards the right. And this happens over and over again, each time you record in the lateral entorhinal cortex specifically. As shown here, but it raises one question, and is this a reflection of time? Is it clock time or is it some other time? And it turns out that it is not really a reflection of clock time, because each time something important happens, then there is a bigger jump in the state space than when it's just nothing is happening. And that can be measured by so the hypothesis is then when something important happens, when an event happens, often what you call an event boundary, then there's this little jump. So for a rat, that could be when it walks in a maze and suddenly comes to a reward. That's a big thing for the rat. And then it may move around. Next time it comes to reward again. And at those points, we hypothesize that perhaps there may be bigger jumps. And that's what it shows. So if you now, just for the sake of time, focus on the right plot, then you will see that when the rat comes to reward, the speed of the trajectory in the state space increases, and then it decreases again. So this is actually the change in trajectory speed. So this is acceleration, and then of course a deceleration, and then it goes back to the same speed. I skip that one as well. But let me then just sum up this first part of the time section here, which is that we have a drift in the activity in the lateral entorhinal cortex, partly reflected also in the hippocampus, that expresses the passage of time, which is very different from what you see, at least in the medial entorhinal cortex, where the activity is fairly stable. This is not continuous, not linear, but it goes in jumps each time something important happens, so that you can also imagine the passage of neural activity is discretized into small blocks of time that are defined by when important things happen, or quite often when there's a change in context, when you get into a new room, when you leave the room, when, for example, I started my talk, when I end my talk. So these are typical event boundaries, and that's where we would expect in a human brain these bigger changes to occur. So what is stored in memory then are these blocks, and then when you try to recall how much time has passed, you use those blocks as an estimate of the time, because the human brain and the mammalian brain is not good at estimating time, but you can infer it from the events that you have stored in memory. So let me then also say a few words about another aspect of time, namely the brain's ability to sequence activity, to store orders of activity in the brain. Because all our experience is a sequence of things that happen, and our brain needs to form those sequences, just like the brain must be able to make a movie that is stored in the brain, not only still pictures. So how does it do that? And we addressed that question in a way where we actually tried to isolate the brain from sensory inputs. So this is the work that was first started with Flavio Donato, who is now in Basel, at the University of Basel at the bottom, and then taken over by Soledad Gonzalo Cogno, who's done most of the work for the last 3-4 years. What they did is that they used the data from mice that were running head fixed in the same position but running on a ball, not moving forward, but doing that in an environment that was completely dark, no sensory inputs, and there was no rewards, nothing. They just ran when they wanted, and they stopped when they wanted, and they ran when they wanted, and did that for an hour. And then at the same time, we imaged the activity using a different way of recording neural activity, namely calcium imaging. Oops, sorry. We'll start again. And then you can actually see the cells that are active, so they blink each time because they express fluorescent calcium. So each time the cell is active, you see it blinking, and you can see that then in a field of view where you have all the cells lined up with each other. And the nice thing is that this entorhinal cortex, which is in a way quite inaccessible because it's at the back of the brain, between at the end of the cerebrum, and here you have the cerebellum. And you can then set a prism in between and actually image from the surface without damaging the entorhinal cortex at all. And this is then what you see. And then we asked, if the mice run in this very reduced situation with no or very little input, can we then see any traces of any basic sequence activity? And first of all, we found that under those conditions, many, or actually most of the cells in the entorhinal cortex, in the medial entorhinal cortex, oscillated at the time at the frequency that was extremely slow. So as you can see at the top left one, for example, this is the autocorrelation of one cell, and you can see that it's correlated with its own activity at periods of in the order of about 1 minute. So it's extremely correlated with activity 1 minute before or after, and another 1 or 2 minutes and 3 minutes, and so on. And the same for the second cell, and for the third cell is more noisy. But if you line up all the cells, you get what you see in the color diagram here, which shows time on the x-axis, neuron number on the y-axis, so 400 plus neurons. Each row is 1 neuron, color indicates activity. And you can then see that they all have these oscillations being active at approximately intervals of 1 minute, and the whole network enters this mode when the rats mice run in this protocol. The same you can see when you do neuropixels recording, so it's not dependent on the technique. And then the second part of the observation, which is the most striking one, is that within these oscillations, the cells actually line up in repeating sequences. So what you see here is on the x-axis time over 1 hour, and then on the y-axis neurons again, so some 400 plus neurons. And now they are organized according to when they fire. So it simply shows is organized according to correlations between the cell's activities. You take one starting cell, a seed cell, and then organize them from bottom to top according to how much they correlate with each other, and then you get through the whole sequence. And then it's, as you can see, it's actually circular. When it gets to the top, it starts at the bottom again, and then just goes round and round and round and round through the whole population, where each cycle then takes about a minute. You can do that by the correlation method. You can do that with the principal component analysis, many different other dimensionality reduction techniques. You can do it on the raw signal. You can do it with neuropixels probes. And you can, if you break it down to the key dimensions, the more you see that it's actually circular, just going round and round and round, regardless of which method you use. And at the period of the mean is approximately 40-50 seconds, but it's a wide distribution going up to several minutes. And this is can go on for as much as an hour. Often it's interrupted and then stops for a while and begins again, but it is not related to the behavior of the animal. So for example, you see when the rat stops, so it's running only in the blue area, and the rest is just sitting there, and the oscillations and the sequences still go on. So this suggestion, this weird phenomenon, is probably not what you see when you record from a mouse that is running around in an open environment or a natural environment, but it is something that is revealed when the mouse is in such a stereotypic situation. And one may then ask, of course, what is this good for? And we think it is good for quite a few important things. Namely, it may serve as a template or a scaffold or an internal archetype sequence that is there, a default in a way that is running when nothing else happens, and that can be used to build new sequences during experience on the fly when it's needed, and you have to store it in memory. You can't really invent sequences each time, but it's very convenient for the brain to have such sequences, and you can just then recruit them, use them, and combine them in different ways, one level down, and you can much more easily generate new sequences, which you have to do when you want to encode memories that are sequences by nature. So finally, a few words about microscopy. So I mentioned that these mice in the last study were head fixed. They had to run under a microscope, which is not really ideal if you want to study natural behaviors. So together with Wei-Jian Song, who came from Peking University to the lab about 5 years ago, we have developed a new version of two-photon microscopes that are different from the one you saw in the sense that they are portable. They can be carried on the head. They're only 2 grams of weight, and they still have the resolution similar to the big benchtop microscopes. So the classical way of recording has been using these head-fixed mouse preparations where they run in a virtual environment. So you make it like a cinema, and you simulate moving in space, but they don't do it. They don't have vestibular inputs, and it is a quite deprived situation. So for that reason, people have for a long time wanted to record from freely moving mice. That has happened for about 10 years with so-called one-photon microscopes, which have much less spatial resolution and lots of background fluorescence and no resolution in the C-plane. But people have really wanted to develop two-photon portable microscopes, and it's a 20 years history of that. But the problem has been over the years that many technical limitations. The calcium indicators, first of all, weren't good enough in the beginning. Scanners were too slow, too small scanning angle, and so on, leading to microscopes that were far too heavy to carry for a mouse. And the new thing here now is the technical development of a miniscope, only 2 grams, that still can image thousands of cells for various reasons that I have no time to show here, but you can see the behavior of a mouse carrying such a miniscope in the middle column here. So it's indistinguishable from a mouse that has nothing on the head, whereas one of the old ones, you can see how easily they get tired and just walk around the corners before they sit down. So it's a major change in the quality of behavior that you can address with these miniscopes. So here just some recordings from a mouse that is running to the right in a box while it's searching for food crumbles or chocolate pieces, and you see the activity in 4 different planes at the same time. Here you have another recording, a mouse climbing a tower, a vertical tower, and which it does quite easily, 20 centimeters up, to get a biscuit with vanilla, which they really like. And then they eats it, and then it will even jump down and do the same thing 11 times. And at the same time, you see the activity to the right, where the motion artifacts are actually so small that you can barely, it's not more than you would have in one of these benchtop big microscopes before. So it's a big change, allows us to study the activity of the cells in the entorhinal cortex and elsewhere. And this is just an illustration of where grid cells are compared to object vector cells, and on the work of Horst Obenhaus together with Wei-Jian Song. And it's just, of course, the interesting thing is that not only can you record the cells, you can know which anatomical cell types they are, where they maybe project, and you can stimulate the same cells too, so that you can actually probe the connections and influence the activity in the network. So no time to say more about that. Just summing up, I had one part of the talk was about the spatial basis, or the neural basis, neural computation of space. Another one was about time, which is much more recent. It's more difficult to study, much too fast, of course, so it's harder to study, but there are some interesting developments in that field as well. And finally, 2 slides about the obvious link to disease, namely Alzheimer's disease is a disease that starts out very often with damage in the very same area of the cortex that we have been studying, the entorhinal cortex, and especially the lateral entorhinal cortex, and then goes to the medial entorhinal cortex, hippocampus, and then goes all over the brain. Those changes actually happen often decades before symptoms are tested, which is, while there is no cure for Alzheimer's, it's also good news in the sense that when a cure comes, then actually can be addressed. The time window where you can actually interfere is quite long after all. And the fact that it begins often so early in these brain years explains, of course, why spatial disorientation, confusion about time and location are key symptoms of the early stages of the disease. So no need to tell anyone how prevalent Alzheimer's disease is, and that it's getting more and more prevalent because people get older. Life expectancy is shown to the right here, just from 1998 to 2015, increasing in most countries by 3 to 4 years. So it is an important problem to address, and the fact that we are doing basic research in our community is again an example of how you can actually have something available that you can apply, just like for the COVID vaccines is another illustration quite recently. So with that, just mention that of course I didn't do this alone. There are lots of people who contribute. I mentioned most of them, I hope, on the way. May-Britt, Moser participating in all the work. To the right, you have names of some of the early people, and the funding is listed to the bottom left. And with that, I think I've spoken far too long, but I can stay a few minutes more and take some questions. Thank you very much.